Yes, so I will be covering molecular biodesign and also the bioinformatics. But first, I will start with molecular biodesign. Here is my contact details here if you need them. But of course, Andrea will no doubt share them at the end of the presentation as well, or in some kind of follow-up. So what is molecular biodesign? Molecular biodesign to me is, I think, a very fun process because it's a concept of understanding biology and then from that understanding engineering something new, for example. So we understand what happens naturally inside a cell, a bacteria, a yeast, humans, and then think, okay, how can we change this so that we can then apply it to maybe a different situation or make something better? So it can be applied into industry, healthcare, or maybe conservation of the planet, for example. So the aim is to create new biological systems, new properties that can make the world a better place, improve healthcare systems, things like that. So in this quick example, we have like making environmentally friendly biofuels, targeted cancer therapies, or agricultural solutions. And those are completely different things. And that's what I think is also exciting about molecular biodesign is that it's so diverse. And if you have an interest in agriculture or healthcare with humans, then this program works for both of those people. An example of this, to put it into context, is in the 1950s, for example. Everyone, I assume, is aware of diabetes and the need for insulin. How they used to get insulin for people with diabetes is that they would grind up lots of pig pancreases from slaughterhouses and try and isolate just the insulin to then give to people with diabetes. And you can see the huge number of pig pancreases that are needed to just produce a tiny amount of insulin. And I think you'll agree here that doesn't look very efficient. So instead, this is where molecular biodesign comes in. The gene for producing insulin is easily added to a small biological DNA piece called a plasmid. And then we can insert those plasmids inside of E. coli, a simple bacteria that's really easy to grow. So we have a protein chain B and a protein chain A, and we put them into different E. coli. And then we keep them in these vats, these big vats here, and we're just growing lots and lots of E. coli. And then through processes of harvesting, we just harvest the protein of insulin, and then we have loads of it to use. And no pigs were harmed in the creation of this insulin here, and you can keep this going forever and ever and ever as long as you grow your bacteria, which are very easy to keep. And this is understanding how biology works. How does insulin work, for example? How does it work in the genome? And then making bacteria do the work for us and just harvesting the proteins from the bacteria, which is much more efficient. So it's processes like this, but it could be insulin, it could be glowing genes, for example, that make things glow in the dark, perhaps not as useful as insulin, but it could be other drugs as well, if we want to make other drugs or making enzymes that make can maybe fix oil spills, for example. It can be anything else. If you're a student and you come to study molecular biodesign, it's a very broad field. As I said, you could be working with cancer therapies or you could be working in agriculture in the future. And we've tried to have an education that's equally as broad. So you will learn about biological processes, biological organisms. What's the difference between a plant and a human, for example? It's easy to see, but what's happening in the genome? What's happening there? Maybe about growing different kinds of cells. There'll be quite a bit of lab work as well and data analysis, as you can see from these graphs and the connections maybe of genes. And it's all about understanding how does the biology work and then how do your new systems work or are they efficient in producing proteins that you're interested in. This is how the program looks. So it's a similar setup as Araceli was discussing. In year one, it's very much a foundation, so we're trying to bring everyone to the same level. And we share many courses with biomedicine, for example, but we have some differences as well. We have a color code system here. So the oranges are chemistry and blues are bioscience focused, so a bit more biology focused. And the yellows are more bioinformatics, and this is using computers and data analysis. And then the darker the color, the more complex the course is. So it's a higher level. So in the first year, you're learning the basics about evolution, microbiology, genetics. We also have, of course, general chemistry, the basic level chemistry to understand the next step. Year two, we're looking more about specific processes that can happen within the lab, doing some genetics in the lab as well. And then we're moving towards biochemistry, which is a bit more of a specialist chemistry area in regards to biology or biological organic molecules. We also will teach you how to write papers as well and how to write research papers. In the second half of the year, you're going into deeper into Python, and we're quite excited to make some changes this year. So this is a new updated program. And this year we will have, when you are joining, we will have Python mandatory programming courses. And this is really good for the future because it means that if you want to go on to further studies, for example, or you want to go into work and you have to work with data quite often as scientists, you will understand how to manipulate this data or to learn even more programming skills. So programming skills for biologists are really in demand. So this is why we've included it into this program. We have molecular biodesign, where we will make glowing organisms. And then in the final year, you're really going up an extra step here. So Molecular Biodesign 2 is a fantastic course that has CRISPR, genetic modifications. And CRISPR is in huge demand, and maybe you saw it won the Nobel Peace Prize a few years ago. And this is something that our students really enjoy this course because it's a big one, as you see. It takes a lot of time and it's a lot of time in the lab, but it's also really rewarding and fulfilling. And you're working with real higher end science here. We're going to also have protein biochemistry. So you've got a lot of chemistry, but this is good for the future. And we also have a second eligible course section here. So these two eligible course sections, you have the choice of specializing. So maybe you're interested in infections, maybe more human science here, or maybe you're interested in conservation, or maybe you want to specialize even more in programming. Then you have the choice of choosing between your specializations. And then we have a second section here when again, you can choose something maybe more ecology based or thinking about sustainable practices, or you can choose to advance your bioinformatics skills and your programming skills one level further with another Python course. Finally, your program finishes with a thesis project. And it's similar to what Araceli was saying, that the thesis project is a time when you have your own project and you will work with whoever you find really in an area of interest that you're interested in. So it can be at this university, but it could also be at Karolinska, it could be in Stockholm, it can be abroad in other countries. You can email companies and institutes to ask, could you take a thesis worker and work on whatever they are doing? So it's a really good opportunity to develop skills and be part of a project that you are really, really passionate about. And you can do that here or abroad. So just as an example of how broad this area can be, and you can choose your own path, I suppose, in that sense, there's some thesis workers from previous years. So we had a student investigating fertility of the human ova, the IVF. We had students testing GMO plants. We had students looking at soil microbiome from nature reserves, looking at inflammation and cancer, and also looking at detection possibilities for, I'm going to struggle to say this, dermatophytosis. Oh, I did struggle to say that. But as you can see, it's very, very diverse. And that's super fun for me because you can go in areas that you think is interesting. Our students in the future, they also have diverse career goals. Some of them work for private companies such as Eurofins doing testing, AstraZeneca doing lots of research, drug development. Some work in uni labs, more clinical settings in hospitals, for example. Many of our students do this. And many of our students also go to do further studies or to work as researchers at Gothenburg, Karolinska, for example. Prerequisites, I think Andrea will talk a little bit about that later, so I'm going to skim over this here. Um, yes. And we also have the studying abroad also. So you're also welcome to study abroad and encouraged. It's a really fun opportunity. I know you might be studying abroad anyway, but you can study abroad for some parts. And I believe there is funding available. And it's, again, another opportunity to expand your network, expand your horizons, have an adventure. And we have some common partnerships that you can follow up with, but you can also find your own as well.